Welcome to another episode of Inside UMiami Medicine. I'm your host, Henri Ford, the Dean and Chief Academic Officer of the University of Miami Leonard M. Miller School of Medicine. In our last episode, we explored promising advances in the treatment of advanced deadly brain cancer. Today, we continue our focus on glioblastoma, diving into groundbreaking research that uses viruses to make cancer cells more susceptible to treatments. This is a rather novel and fascinating approach. And to help elucidate and discuss this truly outstanding and wonderful new therapeutic approach to address this vexing problem, I'm thrilled to welcome to the studio Dr. Ashish Shah, one of our distinguished neurosurgeons and superb brain cancer researcher. Dr. Shah is an assistant professor of neurological surgery and the director of our neurosurgical oncology fellowship. He's also a quadruple K, and we'll hear more about that. And so thank you so much, Dr. Shah, for joining me this morning and to help advance our understanding of novel strategies to treat glioblastoma, which we know is so devastating. And that's been a primary focus for you. But before we delve into our topic du jour, I'd love for you to share with the audience a little bit about your background and how you got to become such a prominent neurosurgeon and researcher.
Sure, yeah. It's kind of a long story. I originally grew up in Texas, South Texas, Corpus Christi. I don't know if you're familiar with Selena from the movies, but that was our hometown. And I grew up my whole life there. My parents immigrated from India around the 1980s, and then we set up shop there. And it was around 17, 18 years old. I was looking at these kind of BSMD programs, the baby doc programs, if you may say. And University of Miami had this great one called the Honors Program in Medicine. And I was looking around the country, and it was January. It was a nice 70 degrees here. And they had me come out here for interviewing, and the other places I went to were negative 20 degrees. And this was an easy decision to come. And when I got on campus, I was immediately in love with the university, and I haven't left since. It was just an amazing time, just kind of being here.
And what led you to study neurosurgery in particular, and then that brilliant career in research?
This is a great question. Actually, it all kind of leads back to Haiti, actually. So when I was an undergrad, I really was interested in global health and kind of mitigating health disparities on the global scale. And I heard about this program called CaneShare. It was the medical school sister of the Project Medishare in Haiti. And it was led by Dr. Barth Green. And I reached out and said, hey, can I come help out? I was like, I can take blood pressure, I can take vital signs. I can come help out. And they said, yeah, sure, come on our trip. I said, okay, sure. So I went down there. We went to Tumor, the Central Plateau in Haiti, and we were taking blood pressure and prescribing, anti-parasitic medications in the Central Plateau. And I met, Dr. Green there, and I said, I was like, what are you doing here? He's like, I'm chairman of neurosurgery. I said, well, so why are you here for, prescribing blood pressure medications? He's like, this is my life. And I was like, when I grow up, I want to be just like this guy. And so when I came back, I just started shadowing him in the OR, and it became, it became a passion of mine, looking at kind of bridging, the scientific gaps in knowledge, and then global health, and then, neurosurgery, and the rest of the pieces kind of fell in place.
It's this fascinating story. A brilliant neurosurgeon treating parasitic infection, and then that gets you involved, that gets you excited, and you become a neurosurgeon yourself. So where does the research interest come in?
So, neurosurgery is one of these fields that is, and forgive me, I'm going to bring another Haitian proverb in here, mountains beyond mountains. There's a famous Haitian proverb, and so neurosurgery is exactly like that. Every single time you do encounter any type of surgery or research problem, there's always another hurdle to kind of overcome. And for me, that's what neurosurgery was, and the scientific inquiry in neurosurgery. So I felt that, that this was kind of melding, two passions of mine, kind of scientific curiosity and the clinical aspect of just challenging difficult cases. And so it always pushed me to kind of to kind of go and beyond the next step. And in research in neurosurgery, there's so many gaps in knowledge. We don't know so many things about the brain. And for brain tumors particularly, which is my clinical interest, our therapies have really not worked. And so for me, it's that, what can I use my skill set for? And I felt this was the most pressing need.
And how did you prepare yourself to begin to address some of those questions? How did you begin to sharpen your research focus?
Absolutely. Yeah. I think getting involved in research at the early stage was really, really critical, and finding the right mentorship, because mentors are the ones that kind of give you the ideas and allow you to kind of pursue the important questions you have. I started in undergrad, working on kind of infections in the environment. And then, in medical school, I kind of started working a little bit more, looking at kind of easier, simpler problems. But in residency is really where I took off my research significantly, and I was able to work with virology experts, Nori Kasahara, who is here at the University of Miami, and then I went a year to the NIH to really sharpen, my skill set.
And what made you decide that you needed to go to the NIH?
So yeah, the University of Miami, neurosurgery program, you learn to do every type of surgery under the sun. We are one of the highest volume centers. And clinically, I felt like I could do everything, and I had a great basic science background, from medical school and in residency. But I really wanted to bring clinical trials to our patients and kind of really bring the translational aspect, because it's great to do everything, in the lab and the petri dishes and this, but can I really bring that discovery to our patients and kind of change outcomes in a rapid way? And I felt like at the NIH I would be able to make some connections and kind of really bring that back home.
And that's what that year did for you.
It was a great year to kind of make new connections, learn a skill set that I otherwise didn't have, and I was able to kind of bring it back down here.
Okay. Now, you've chosen to focus on the most difficult cancer to treat. Tell us about the rationale for it. Is this a high risk, high reward approach, or is it that some of the tools that you've acquired make you more optimistic about your ability to find a sensible cure for them?
Absolutely. So glioblastoma is one of the most deadly cancers. The outcomes have not changed over the last 25 years, despite, all these advances in immunotherapy.
If you remind the audience about how deadly it is.
Sure. So, from the time of initial diagnosis, overall survival is around, a little bit over a year, meaning that around 50% of patients will succumb to their disease in over a year. And that's despite our maximal surgical efforts, radiation, and chemotherapy. And so for a surgeon, that's quite devastating. You're providing your 15 years of training to take this tumor out of someone's brain, and then you're giving as much treatment as possible, and to know that it's not good enough. And so I said, well, what can we do? What can we do to really improve these outcomes for our patients? Are there ways that we can harness the immune system? Are there ways that we can use biologic therapies like viruses to kind of really improve our responses? And so
viruses, where did that go from? Help me make the connection for us.
Yeah, no, it's actually a great, I'm a history buff, so it's actually kind of a great story of viruses and cancer really have been, interlocked, and we've, since the early 20th century, when we discovered the viruses can actually cause cancers. And it was really a retrovirus that was the first discovery that these viruses can actually cause a cancer initiating cell.
And they cause a mutation.
Yeah, interesting. Yeah, they have these genes within the actual retrovirus that are activated. There was a story about a chicken avian virus that basically can be passed on from a fowl to another fowl, and that was the first discovery by Peyton Roos in the 1920s. And so this discovery kind of led us to discover what the retrovirus is. And there's been a link between these retroviruses and cancer development. And this was kind of sharpened as the HIV pandemic was going around. And we've kind of linked, these, certain viruses can cause cancer, and the WHO estimates that around 20% of cancers are linked to viral infections.
So now guide us.
Yeah.
How are you employing this knowledge to address our difficulty in treating glioblastoma?
Sure.
I'm a pediatric surgeon, so that's what I know.
There's some research in neuroblastoma as well coming out, but Yeah, I mean, knowing that 20% of cancers could be related to viruses, my question was fundamentally, is there some viral etiology or some viruses that maybe cause us to have a higher risk of glioblastoma? And there's been some work done in this before. My interest is in something very overlooked, which is called the human endogenous retroviruses, which are otherwise called HERVs. But these are basically ancestral evolutionary relics of viral infections that came into our germline, millions of years ago. So in layman's terms, essentially, it's like viral infection happened 6 million years ago in human beings,
and we still have relics of those infections. So they are incorporated in our genetic makeup.
Right. So you and me, about 8% of our genome is made up of these viral elements. Now, everybody asks the question, are these infectious viral particles going to go from one person to another? And in general, no. These, our bodies found a way to use these to allow us to, during development of our embryo, kind of helps us out. But then afterwards, when we're normal, adults, these viral proteins completely go away. But in cancers, they start making an appearance again.
And how do they help trigger?
Yeah, so this is a very interesting question. So a lot of the work has been suggesting that these proteins from these viruses actually cause the cells to become more stem-like. So they become, resistant to treatments. They become more pluripotent, so they can kind of morph into whatever cells they want to become.
So the cancer cell now we're talking about.
Right. So it's in the embryo, when they're in the 8 cell stage, you can actually see these viral proteins being, in the blastocyst, you can actually see these proteins being expressed. Now they go away after differentiation, and then in cancer cells, they start making these proteins again. And we found these exact viral proteins and particles on glioblastoma stem cells.
Wow. Okay, so now, having this knowledge, how are we going to target them to have a better outcome?
So we take a little book from the virology playbook. So we know that these are retroviruses, and we have drugs to treat retroviral infection. For example, like HIV medication. And so a lot of work done by my mentor at the NIH showed that antiretroviral therapies like reverse transcriptase inhibitors can actually reduce the levels of this protein and RNA in cancer cells and in normal and in other cells. And so can we use these drugs to target this glioblastoma viral protein?
So is the strategy then to make, by using these antiretroviral drugs, are we making the cancer cell, the glioblastoma cell, more susceptible to the treatment modality that we use?
That's the idea, is that these cells are, these glioblastoma stem cells per se are the ones that are, responsible for recurrence. So if we can add on these antiretroviral therapies onto our standard of care, can we really improve outcomes? And so, with some help with one of my collaborators, Dr. De La Fuente, we're actually starting a clinical trial soon where we're going to be adding this on for one of the most difficult to treat unresectable glioblastomas, where we're adding on antiretrovirals plus chemotherapy and radiation.
Okay. But that's not quite gene therapy in the classic sense.
Right. So one side of our lab is kind of looking into kind of what viruses can cause cancer. Another side of our lab is looking at, can we use viruses to kill cancer?
Isn't that somewhat paradoxical?
That's right.
On the one hand, they're causing, on the other hand, we're using them to target the cancer cell.
Correct. So
tell us about that.
Yeah. So, we're, the idea, is that, these cancer cells are very, very heterogeneous, and they're a moving target. And so to attack a moving target, I think you need some sort of biologic therapy. There's another moving target. You need a moving, so in terms of, we're using retroviruses to basically attack the glioblastoma cells and insert a gene therapy into the cells to make them more susceptible to certain treatments. And so this has been kind of looked at for about 2 decades in cancer research and clinical trials. And one particular retrovirus, this called DB107 is the one we're looking at, is basically able to go in selectively into glioblastoma cells and not infect normal cells. And so the idea is that hopefully we can insert a kind of a gene therapy within these cancer cells and then hit them with an oral drug that will kill only the cancer cells and protect the normal cells.
So you have to walk us very slowly.
Sure.
On the one hand, we have some viral components in our genetic makeup that may go back millions of years. We think that they may help trigger the development of the glioblastoma. Now we want to use yet another form of virus to go attack the cancer cell. And we were going to use antiretrovirals to actually make the cancer cell more susceptible. So how is it that this viral-based gene therapy is going to do something counterintuitive? Because we were trying to target the viral particle.
Right. So this is a completely separate virus. The one we're looking at that we think is contributing to the what we call cancer formation is from a human origin. So the other virus we're doing is basically genetically engineered virus made in the lab. It's been tested in clinical trials as safe. And basically what we do is with that virus then is a replicating retrovirus. So it actually replicates only in the cancer cells. Our normal cells actually don't get infected by it because they're in the brain, they're not replicating.
Very good.
And so it goes in the cancer cells, replicates, and it sits there dormantly because it's a gene therapy. And what we do is we give actually an antifungal drug, which doesn't do anything in the normal body except kill funguses. But because these viruses have been infected with a fungus gene, we actually kill only those cells. And they take it by a pill. So you can take this pill by mouth and minimal side effects. And so we actually started and enrolled our first 2 patients for this clinical trial for recurring glioblastoma.
So fascinating. So you are capitalizing on the fact that the glioblastoma cell is dividing, and by targeting in that, then you can use that whole viral apparatus to outsmart it and make it more susceptible to
absolutely, yeah,
to therapy. Okay.
Now we're hoping that because it's replicating, that it's able to kind of go with the tumor where we can't see. And we're using some novel approaches to kind of deliver the virus within the brain itself through catheters and allowing the virus to really permeate in the areas we can't reach with normal surgery. And
it's going to obviously improve sensitivity to whatever treatment that we give it. And that's what you're exploring with Dr. La Fuente.
Absolutely. And so the really unique thing is we actually developed a biomarker test here at the University of Miami where we're able to look and see which patient is actually going to benefit the most from this virus.
That's what I want to ask you. So is it widely applicable? Are all glioblastomas the same? Or are there different subtypes that may be more susceptible to gene-based, to viral
Yep, that's a great question.
Therapy.
So South Florida is such a diverse patient population here. And so what we found was that there's a specific biomarker that's really enriched in this community. And that biomarker in clinical trials showed patients benefiting quite a bit from retroviral gene-based therapy. So we're screening patients for that biomarker and then enrolling those patients. So we have the highest chance of success.
Okay, so you have a clinical trial. So tell us a little bit about that and where you told us about enrollment, who qualifies and so forth, and what you hope to see.
Sure. We're enrolling hopefully around 30 patients with recurrent glioblastoma who have this biomarker. And we're employing something very different. It's called what I call adaptive virotherapy, which means that, in normal clinical trials, you go and inject one time the virus and then see if it works. And if it fails, you say, okay, this didn't work. But a lot of what we're learning from Japan, from our colleagues up north in Boston, is that repetitive injections allow the immune system to get primed to the virus and virotherapy and allow the virus to really work. And so we're actually doing something where if the virus has escaped our viral-based gene therapy, we actually inject it again. And so we're allowing the virus to get to the cells we can't see. And so I think this is where the emerging treatments for glioblastoma are really going to have the most effect.
And any preliminary observations so far?
It's early. We just opened it up, but I'm very optimistic. Everything's been so far safe, and we're pretty optimistic.
And who's funding this project?
The National Cancer Institute was able to give us a R21 to fund this, and also the university has actually been very generous with supporting these investigator-initiated trials. So, thank you again for allowing this to.
This is exciting. If you can find an answer to this vexing problem that affects so many people. It would be a major breakthrough. So we are just glad that you're here and pursuing this work along with your entire team. So which antiviral drugs, man? So we're looking at two approaches, right?
Right.
This antiretroviral and then the gene-based therapy. So I'd like to know, A, what antiretrovirals are you using? And then B, of those two approaches, which one do you think holds the most promise?
Yeah, I think that's a great question. I don't want to put my bet on either one yet, but we're using.
That's why you do the experiment. They're hypothesis driven. Okay.
Correct. I do think that the antiretrovirals do make the tumor cells more sensitive to chemotherapy and get rid of these stem-like cells. And the benefit is they're able to go to the brain at very low concentrations. They're safe. We've tested them in so many clinical trials. Patients are able to tolerate them with minimal side effects.
Okay.
So it shouldn't add to the burden of chemotherapy. And we're using two specific ones. So just like HIV, you treat with multiple medications because these retroviruses avoid drugs and they have drug-resistant mechanisms. So we're using reverse transcriptase inhibitors, two different ones, and a protease inhibitor that is actually found to make cells more sensitive to radiation. So we're adding in, kind of three different medications on top of standard of care.
Well, and when do we anticipate having some preliminary results to share?
So we're hopefully getting our IRB. We've received IRB approval, and so we're hopefully going to recruit our first patients in the next 2 months.
And how do you get that virus into the glioblastoma again?
Right. So it's unique. As a surgeon, this is our life. We, everybody asks the question, oh, well, how can you get this drug to the brain? I said, my friend, I see the brain every day. So how we can get it there. I can get it there. When we resect the tumor, we can inject very slowly the virus, with a TB syringe actually into the tumor cavity. But, a lot of data suggests that you don't really get good diffusion or basically penetration if you do that just that way. So we combine that plus something called convection enhanced delivery is a very fancy word.
Let's break it down.
It's a very fancy word to basically saying we're slowly infusing our virus into the brain and that allows you to get a much higher volume of brain infused with the target of what you want to get in.
Great. So we touched on it, but I just for some clarity, because some people are going to be listening out there and they say, wow, maybe my family relative should be a candidate for this therapy. How are we selecting patients?
So we have kind of strict inclusion criteria. So for the antiretroviral therapy trial, that's for newly diagnosed glioblastoma. So before you've actually gotten treatment, we actually, and this for unresectable tumors. So tumors that we can't surgically access to remove, we actually perform something called the needle biopsy and the laser ablation, which is another way we're able to kill the cancer cells minimally invasively. And then we add on the antiretrovirals. That's the antiretroviral trial. The gene therapy trial is for recurrences, so after you've had treatment.
Fascinating. Are there any other centers doing similar work?
We're the only center currently recruiting and enrolling for both studies. So it's very lucky to be here and conduct such.
This is the place where the major breakthrough in the treatment of glioblastomas may occur because of you and your team.
I hope so. We're very optimistic, and the team here is we have one of the highest volume brain tumor centers in the country, and because of that, we're hoping to really change the paradigm.
That's fantastic, and it's great to know that you have given me and so many others something to hope for, because we know how badly this disease how badly it affects so many lives and destroys families, and so we would love to see something happen.
I appreciate it, Dean. It's a passion of mine, and I think every day we get to work, our lab is committed to changing the narrative.
Yeah. In the remaining few minutes, I want to go back to the genesis of your interest in neurosurgery, which occurred in the Central Plateau of Haiti. And And I'm curious how you're continuing to link this amazing work that you're doing in advancing novel therapies for deadly brain cancers to some of the more pedestrian needs of the Haitian healthcare system in general, and relating to neurosurgery in the country. So
that's a fantastic question, and this is something that's really kind of plagued us, when we started getting involved, early on. And I was in medical school, and we kind of did a survey of Haiti. Said, how many neurosurgeons are there for a country of 11 million people? And it turned out there was about 4 neurosurgeons, and out of them, maybe 1 was completely trained in and so one of them was actually the prime minister or the president, so really, there was a deficit in training. And so how do we really correct this? And the University of Miami actually had the first family medicine residency program that started in Cap Haitian a long time ago. And so we asked them, can we take a page from your playbook and kind of really, start a neurosurgery residency program? We have the tools. We go down there often. We have a hospital, Bernard Mebs, that we work with closely that is training, nurses. And can we use this infrastructure to really develop a neurosurgery residency program? And with their support, we wrote a complete curriculum under the Congress of Neurosurgery's kind of guidelines. We went to the Haitian Ministry of Health with John Ragged, Barth Green, and got it approved. And then we started training our first resident.
Amazing.
And he's a fully trained neurosurgeon now. He's
amazing.
He's training actually the next generation.
So it's train the trainer. So you are increasing capacity.
That's
really what it's about.
Going down as a mission is not going to solve any problems. So
that's fantastic. And in your research in terms of neurological disorders down there, what are some of the prevalent needs, and how are you helping to address some of those?
So you can imagine
in your spare time.
Yeah. Well, we have a lot of help, so I can't say it's all me. I think really the main thing in developing countries is that brain tumors are important in terms of research for me, but the thing that is the highest cause of morbidity and mortality are trauma and hydrocephalus, actually. So it's a pediatric disorder where children could have an infection at birth and develop a fluid in the brain and really causes long-term morbidity. And as neurosurgeons, we have tools to fix those by doing a very simple 30 to 40 minute procedure where we divert the fluid away from the brain. And so our research now is, can we identify what's causing the infections in these infants? And can we can we figure out how to prevent that? So we're actually working with a lot of different collaborators here at the university to try to figure out what bacteria and what viruses may be causing these infections. So
this is fantastic. And you get to go down there periodically?
Yeah, so
when it's safe.
Yeah, this has been, last couple of years have been tough to go down. We're actually trying to plan a trip hopefully next year to go down once things calm down and there's a little more stabilization. But luckily with WhatsApp and a lot of these Zoom connections, we're able to kind of get real-time information of what's going down with Dr. Lafortune, who's our resident down there. And he basically tells us, about cases going on, asks for advice. And so right now that's been sustainable, but we definitely want to get down there and, get our feet wet again.
Fantastic. Well, Dr. Shah, it's just been an amazing half hour of discussion with you. And we're so fortunate to have someone of your caliber and compassion on our faculty. So it's been a real joy to talk to you this morning.
I appreciate it. Thank you so much for having me.
And this is just the beginning, because we're going to want to have you back to give our audience an update on the progress of your antiretroviral and viral-based gene therapy to address glioblastoma.
We have a lot of work to do, and this is the place to do it. So
fantastic. Well, this has been another episode of Inside UMiami Medicine with the phenomenal, brilliant young neurosurgeon, Dr. Ashish Shah. Tune in for the next edition.